Welcome to our today's webinar, Oxygen and pH in Cell Cultures, Engineered and Native Tissue, Visualized in Vitro and In Vivo with the VisiSens system. With me, my name is Gernot John. I'm Director Marketing and Innovation at VisiSens, and with me is my colleague Robert J. Meier. Hi, Robert. First, I would like to say a few words about the company VisiSens. Our company is based in the south of Germany. We were established in 1997 as a spin-off from the local University of Regensburg. The company is privately owned, and we are currently more than 90 employees. The company is certified according ISO 9001 as well as ISO 13485, which is the standard for medical products. We are focusing on measurement systems for parameters that are essential for life: oxygen, pH, carbon dioxide, and recently we added system to measure biomass. We have non-invasive sensor systems that measure through the wall for plastic or glass, as well as microsensors. These are micro-invasive probes that measure in tissue, animals, leaves, and also in every small environment. Before we start with our presentation of the imaging system, I would like to give you some result from the questions we asked you up front about whether you are working with the VisiSens system, and with the VisiSens systems. So from you, 45% are already working with VisiSens measurement systems, and about 10% are working with the VisiSens system. Let me introduce our today's speaker, Robert J. Meier. Robert is working at VisiSens in the R&D department for imaging solutions. Before he joined VisiSens, he was working at the Institute of Analytical Chemistry, Chemo and Biosensors at the local university. He has more than 8 years of experience in the development and also in the application of optical chemical sensors and imaging. Before Robert starts, I just want to mention that at any time during the presentation, please feel free to ask questions through the question module in the webinar software, and we will answer them either in between or after the presentation. Okay, Robert, the stage is yours.
Thank you, Gino, for the warm introduction. I also want to welcome everybody to our today's webinar on oxygen and pH in cell cultures, engineered and native tissue, visualized in vitro and also in vivo with our VisiSens imaging systems. I want to start today's webinar with a question to you, the audience. Have you ever thought about seeing your cell sample or your tissue sample breathing or acidifying? During the next 35 minutes, I want to show you how you can visualize the invisible with the VisiSens imaging setup from VisiSens. I want to start with a short overview on oxygen concentrations that can be found at physiological levels in human tissue or in cell cultures. Starting with the reference value from air, which is 100% air saturation, that equals 160 torr oxygen partial pressure. It drops down in arterial blood to 63% air saturation. Various organs are named here in this table, and you can see that they are between 18% air saturation up to 45% air saturation, depending on which organ you regard. Even in skin, it's pretty variable, so the values for oxygenation in skin can be from 0.5% air saturation up to 22% air saturation. So if you want to, for example, grow your cells at physiological conditions, you have to know the exact values that are present in your culture or in your tissue construct or in your native tissue. Even if you work with stem cells, it's pretty critical because the differentiation of the stem cells is highly dependent on the oxygen supply. I also prepared a short table on physiological pH values that can be found in human tissue, starting with a neutral value from pH 7. Cell culture in blood is slightly elevated from 7.3 up to 7.5. In tumor microenvironment, the pH levels are a little bit acidified, down to 6.5 up to 6.9. And for example, on intact skin, you find pH levels down to 5.5 with a huge variation gap. Also, if you regard wounds, you find pH values ranging from 5.5 up to 7.5. So you see that the range from 5.5 to 7.5 covers the most of the applications for cell culture and also for tissue. Actually, you can also find pH levels that are lower or higher within the body, but in this range from 5.5 to 7.5, most of the action takes place. So how can we measure oxygen or pH levels? In our company VisiSens, we are specialized on luminescence optical sensors. And with these luminescence optical sensors, you can detect these two analytes. The benefits from optical sensors are that you can measure non-invasively through the wall of your vessel. That means we use light for signal transduction, and light can penetrate through transparent walls, such as plastic devices or glassware, and you can detect the signals from the outside with the detectors. Thereby, you do not need to open your sample or do sampling while making a measurement. Therefore, you can also avoid oxygen ingress or CO2 emission from the incubators, so your growth conditions stay stable during the measurement. The sensors are not made out of glass, no, there's no risk of breaking. Compared to electrodes, they have the huge benefit that they do not consume oxygen while measuring. This is not a problem if you work in very big, bulky solutions, but if you have smaller volumes, such as in cell culture, for example, in microtiter plates, this can get severe difficulties. Also, the sensors are non-toxic. And further on, the signal that you record is independent from flow velocity. So you can use them in steady state cultures, or you can also use them in flow cultures. Today's webinar is focused on imaging technologies. With imaging, you can generate thousands of measurement points at the same time by recording one image. So you can generate full analyte maps over your samples. You can compare different regions or regions. You can detect hotspots and see where the action takes place in your sample and where the most severe metabolic processes go on. You can visualize gradients, which is pretty important, for example, in tissue engineering or in spheroid research. You can monitor overtime changes of the analytes. You can do quality control and optimize your cultures in terms of your physiological oxygen values or pH values that you want to have for your growth conditions. Further on, you can visualize reactions that take place, and imaging works in a non-contact readout mode. And it works in vitro as well as in vivo. And you can do all that with the VisiSens imaging systems from VisiSens. The measurement principle of the VisiSens detector is pretty easy. We use a luminescent sensor foil that is inside the cell culture chamber or on the tissue that you want to measure, and you measure it in a non-contact way with the detector unit. The detector unit excites the luminescent sensor foil and also records the emitted light, transfers the signals to a computer where they are processed. The VisiSens system contains three major components. First of all, the hardware, the detector unit, matching luminescent sensor foils, and a matching software. As you can see here in this overview, the systems are color-coded, so the oxygen systems are coded in blue, the pH systems are coded in red, and actually we have a third one for CO2 imaging, but in this webinar I will focus focus on oxygen and pH imaging. Now let's go on to the detector unit. The detector head is a portable handheld device. It's a microscope, a USB-powered microscope with an integrated 1.3 megapixel CMOS chip for recording the signals, a lens for magnification from 10-fold to 70-fold with a manual focus, so you can adjust, for example, your field of view, you can adjust your pixel resolution and make the images sharp.
Robert, maybe I can just step in here for a second. We have already questions. Maybe also one remark again to the audience. So at any time, you're welcome to send us your questions via the webinar tool. And there is already one question that fits to this chart, and this question is about the spatial resolution of the system. And whether it's possible here to measure in microscale. Maybe you can give us some numbers there.
Okay, actually the spatial resolution of the VisiSens imaging system is not limited by the detector unit, it's limited by the sensor foils. The sensor foils for oxygen and pH imaging both have a spatial resolution of 100 microns, so you can measure in the microscale. And for the oxygen imaging foil, you can further remove the optical isolation, and then the spatial resolution gets down to 25 microns. So you can really measure in the microscale.
Okay, thanks. I think that's everything for now. Again, to the audience, please feel free to send questions to us, and we will answer them either during the webinar or in the question section after the webinar. Thank you.
Okay. And as already mentioned, the handheld device is USB connected to a computer. Okay, now let's head on to the sensor foils. The sensor foils are, as mentioned, color-coded. I want to comment on the oxygen and the pH sensor foil. The measuring range for the oxygen sensor foil is 0 to 100% air saturation, which equals 0 to 20.9% pure oxygen. The pH sensor foil, the HP5R sensor foil, has a range from pH 5.5 to 7.5, and there's also an additional sensor foil for the pH detection unit that can measure in the lower pH range. The sensors respond within 30 seconds. The sensor foil can have a maximum size up to 40 times 40 square millimeters, and the number of sensing points recorded in one image is over 300,000. They work in a temperature range from 5 degrees centigrade up to 45 degrees, which covers most of the applications in cell culture and also in tissue. And the sensor foils are compatible with aqueous solutions. In case of oxygen from pH 2 to pH 10, the pH sensor foils are compatible with solutions from pH 2 to pH 9. Further on, the oxygen sensor foil is also compatible with certain amounts of ethanol or methanol. So now let's head on how to implement the sensors in your vessels. Actually, this is really easy. You get the sensor foil as a sheet of 40 times 40 square millimeters. You use scissors or a punching tool or a scalpel to cut it into your desired shape or size. You use the provided glue, put it on the colored backside of the sensor, which is facing the detector, and stick it into your transparent vessel. After the glue is dried, you can already use it. If you're using cell culture, you can, for example, also use better irradiation to further make it somehow sterile, or you can use a minute of argon plasma, for example. You can easily integrate the sensor foils into nearly any type of transparent vessels. Here are some few examples. You can, for example, use the sensor foils as they are, or you can put them into cell culture dishes, or into Petri dishes, or into well plates. You can also integrate them into microfluidic devices, or you can put them on skin with occlusion foil, or you can use any other type of transparent vessel. The sensor foils are also biocompatible. Here's an example experiment of grown cell culture on top of the sensor foil with different coatings, or as the pure foil, and on a tissue control culture control substrate. These cells were visualized with the live dead assay, and you can see that most of the cells are pretty well alive, and it's comparable with the normal tissue control substrate. Now let's head on to the software part of the systems. The software consists of two major parts. An acquisition part, where you can record the images, where you can do sessions, where you can comment sessions, write down your experimental details so you won't forget them. And you can also record time series automatically. And a second part, the evaluation part, in which all the calculating and the analyzing of the oxygen maps is done. Within the software, you can use different pseudo-color representations. You can choose your color chart. You can also adjust your range if you want to see more detailed structures. You can do multi-plots of a time series to see where happens what. You can do one-on-one image comparison, for example, with the first image up to one of the last ones to see what changed in more detail. You can do alpha blending to see underlying structures. You can use evaluation tools such as histograms, or there's also a statistic tool where you can define an ROI and get an average value, as well as the mean values as min and max values and a standard deviation. And you can use the integrated time series profile tool to get a time series plot. And there are even much more functions. So now let's head on how you can visualize oxygen or pH concentration. In this example, the oxygen imaging setup is explained. You place the detector into the incubator, for example, place your vessel with the integrated sensor on top, close everything, and start the software. Once you started the software, actually it's ready to use, and you can take single images if you want. They are displayed here on the left side as small thumbnails, so you see what you already recorded. Or you can use the time drive tool, in which you can set the interval, how often times the images are recorded, and also the total number of measurements that are done automatically. And then you just press OK, and then you can go and grab a coffee, or you can go on holidays over the weekend, and the measurement is taken by the software. Once your images are recorded, you can go to the evaluation mode, and the raw sensor response is displayed in pseudo-colors as a ratio from the red to the green color that is recorded by the detector. You can also perform an easy two-point calibration with the oxygen sensors by taking a region of interest value for 0% oxygen and also one for 100. Once you took the values, you click on Calibrate, and the calibration is processed on all the recorded images in that session. Now you can see that the color chart changes, and the values are now displayed in % air saturation. Now you can do some further investigations, for example, making a set profile out of a certain region within that recorded images. You define a region of interest, and then the set profile is calculated by the software. And you can see what happened over time in here. Okay, before we head on to practical examples, I think there are some few more questions from the audience. Gernot?
Yes, Robert, thank you. There's one question is just related to the software. You mentioned that you can stay at home during the weekend and the system is working. I guess the question is related, can I also be at home longer time? Because how long can you have that time-lapse video, or how many pictures captured?
Actually, you can define, this depends on the interval, how often you take the images. You can define the interval from seconds to hours, and you can also make time series over seconds, minutes, hours, days, or some people also do time series over weeks. Then they choose bigger intervals. The total number of measurements depends a little bit on the RAM of your computer, but it's way more than 200.
Okay. Okay. There is another question, I guess, related to the biocompatibility, which you mentioned earlier. What is about sterilization of the sensor foils?
I already mentioned 2 examples how you can make the foils more or less sterile. For example, we are using beta irradiation, or we are using argon plasma to apply to the foils before you use them in cell culture. But for example, the oxygen foils can also be applied to or washed with 70% of ethanol before using them, or you can also use gamma irradiation for the oxygen sensor foils.
Great. Great, thank you. I guess that's it for now. Again, thank you very much for the questions so far, and please be invited to ask further questions. And now, Robert, let's head on to the examples. I think we're all curious to listen to you.
Okay. My first example in today's webinar is on the oxygen imaging in stem cell cultures. Actually, it's on the spatial and also the temporal oxygen gradients that came across stem cell culture surfaces. Therefore, the foil was placed in a titer plate, and cells were grown on top of the foil and investigated in a time series over hours. What you can see is when the cells grew and consume up the oxygen due to their metabolism, the oxygen levels drop down. And you can investigate that over the time. You can also see that you have the direct media control where no cells are seeded. This is pretty important for stem cell culture. It's pretty important to know the tissue oxygenation because it's really a critical factor for the differentiation of the stem cells. Because, for example, if you use your oxygenation levels close to the physiological niche of the respective culture, you can improve the differentiation of your stem cells. The next example is on hypoxia monitoring over a time course of migrating cells. Therefore, in this blue circle in here, a round spot of cells was seeded, and it was covered with the oxygen sensor foil. The backside of the oxygen sensor foil is not permeable to oxygen, so what happens is that the cells consume up the oxygen that is still present in the culture in this spot, and after they consumed up all the oxygen, they started migrating towards areas with higher oxygen contents. And that's what you can see here in this video. At first, you see the hypoxia generated in the middle as a round spot, and then the spot grows. So now here's the hypoxia, and here you can see that the cells are migrating and also consuming the oxygen again, and the hypoxia that you image grows. Now let's head on to more specialized cell culture imaging. This slide is on oxygen imaging in microfluidic cell culture devices. Actually, I gave a webinar especially on that topic about 3 months ago. You can send me an email request and I can send you a link for the YouTube video, or you can also Google the YouTube video on that. Today I want to focus on that lower part in here, which is on a hepatocyte culture on different chip materials. The first row is on COC chips, and the second row is done in PDMS chips. COC is a chip material that is not permeable to oxygen, and what happens is that during culture, the cells consume up the oxygen, and with time, the oxygen levels within the cell culture channel drop down, and you get hypoxic regions. So you do not have static cell culture conditions. But if you use PDMS as a chip material, as in this case in here, the lower case, PDMS is more permeable to oxygen, and thus oxygen from the side channels of the system is allowed to diffuse towards the cell channel in here, or the cell culture channel, and thereby you can generate more even cell culture conditions. Thereby you can adjust, for example, the oxygenation for your cells, and you can also grow them in a more physiological way. Now let's head on to engineered tissue, or oxygen imaging in engineered tissue. This is really of special interest because in engineered tissue or in cell-seeded scaffolds, many people have the problem that they have hypoxic cores in the inner part of the scaffolds due to a lack of oxygen in there. And this causes cell death and also decreases the possible maximum sizes of the scaffolds. Here in this animation, I can show you a computerized model on oxygen concentrations that are present in scaffolds. The red outer part in here is the media oxygenation, which in this example is still high. And the scaffold is seeded with cells. The cells consume up the oxygen, and there's a balance between the oxygen consumption and the diffusion from oxygen from the media towards the inner parts of the scaffold. So what you get is this hypoxic core in the inner part. But what you also can see is that you have these oxygen gradients over the walls from the scaffold. You can see also that you have these oxygen gradients from the top side, but also from the media accessible sides of the scaffold. And what now researchers from the University of Regensburg did with our smart little device, they placed the scaffold on top of the sensor foil. Again, the oxygen from the media can penetrate from the side walls and from the top sides towards the inner parts of the scaffold, but there's no oxygen accessibility from the sensor foil part. So what you get is a cross-section through your scaffold, or the oxygenation of a cross-section through your scaffold. That's what they recorded in here in a time series experiment. At the beginning, you can see here this outer part is the media oxygenation, so it's pretty high, and here it's displayed as % oxygen, so this is air saturated. And as you place the scaffold on the foil, you see that the cells start consuming up the oxygen, and over a time course, at a certain point, all the oxygen from the inner parts of the scaffold is breathed away, and you get these hypoxic regions. But what information you still got from the image, from the oxygen image, is you still got the media oxygenation, so you can survey the media oxygenation over time during the growth process. You can also survey the hypoxic inner part and the generation of that. And you see these gradients from the media oxygenation over the walls of the scaffold towards the inner hypoxic core. And this is really pretty useful information for engineered tissue, because if you use, for example, vascularized systems or perfused systems, you can check on how good your perfusion within your scaffold is. Or you can also use oxygen-enhancing materials that are seeded within the scaffold, and you can prove that they really enhance the oxygen supply within. The next example is on pH imaging in engineered tissue. Therefore, a piece of artificial skin construct was imaged, or this pH sensor foil was placed on top of the artificial skin construct, and the acidic barrier mantle was imaged that is present on the surface, on the top surface of the artificial skin construct. Here in these results on the left side, in the first row, you can see that the pH values upon removal of the pH barrier mantle of the skin quickly rise up to 7.2 or 7.1. After 14 days of healing, the pH barrier mantle is re-established and the pH values get back to the normal 5.5. My next example is on oxygen imaging of a cross-section in spheroids. Actually, cancer spheroids get more and more prominent in research these years and these days because they are a model for three-dimensional cancer. Therefore, the cancer spheroids were placed on top of sensor foil in a petri dish. They settled down on the sensor foil and they built a half ball-like structure, and thereby you get a direct cross-section through your spheroids. So your spheroids settle down. You see in the cross-section that in the inner parts, the oxygen gets really quickly taken up or metabolized by the cells, and you will get an inner hypoxic core. This you can see here in this video. So the hypoxic core is established. You will still have, again, the media oxygenation, so you have the control of the oxygen in your media, and you see how far the oxygen from the media can diffuse into the scaffold. So you have a direct hypoxia control during growth conditions in the incubator, and you do not have to make pretty tedious staining and investigations afterwards. So you do not see the hypoxia after it happens, you see it when it happens. So now let's head on to in vivo experiments done with the VisiSens setup. My first example for the in vivo experiments is skin imaging of an infected mouse foot. Therefore, the sensor was placed on top of the foot, and the oxygenation of the skin was measured. What these researchers found out was that if you have a Leishmania-infected foot of a mouse, the oxygenation gets higher than compared to the normal oxygenation of the skin. The next example that I want to show you is measured in a skin fold chamber. Therefore, they measured a non-menelaomous tumor, skin tumor, in a hamster skin. Non-menelaomous tumors have the real problem that they are not visible by the eye, by the bare naked eye. But you can see them in the oxygen image. The tumorous skin consumes way more oxygen, and thus is represented with lower oxygen concentrations as visualized in here. And you can also see the healthy skin. The healthy skin has a higher oxygenation. So you can see tumors that are not seeable by the bare naked eye just by measuring the oxygenation of the skin. Let's go on more deep into organ imaging with the oxygen imaging setup. In this experiment, the perfusion of a mouse kidney was investigated. Before occlusion of the artery, during partial occlusion, and after total occlusion. What you can see before the occlusion in the healthy system is a well-perfused kidney with a high oxygenation status. After placement of a small occluder, you see that the perfusion drops down, but it's still slightly oxygenated. And after total occlusion, the oxygen levels get down to 0. My next example is on cortical imaging in a hamster brain. Actually, therefore, the dura mater was deprepared and the brain was covered with a sensor foil. And what you can see in this video is changing oxygenation of the hamster brain with different breathing gas oxygen contents. In here, you can see the artery, and you can see here the venole. You can see a clear difference between the oxygenation of the venole and the artery. But what you can also see is the parenchyma oxygenation and also some smaller capillaries. And you can see that the oxygenation in the brain, maybe I go back down here, changes immediately upon changing the breathing gases. Okay, for my last example, I have an in vivo example in humans. And this was perfusion monitoring of microvascular flaps. In microvascular flaps, you may have the problem that they are not well perfused, and if you put them into the body again and they are not well perfused, they may die over time. So what we checked in here, or what some researchers in a project from the University Klinikum of Regensburg did, they checked the perfusion of the microvascular flaps with the VisiSens system. Therefore, they used a handheld detector, placed the sensor foil on top of the microvascular flap, and recorded in a time series the uptake of oxygen from the microvascular flap. If the flap is perfused well, it takes up the oxygen way slower because there's an inner oxygenation within the flap. If the perfusion is bad, it takes up the oxygen faster. And by this uptake speed, you can define if the microvascular flap is well or badly perfused. So I hope I presented you some interesting results using the VisiSens systems for oxygen and pH imaging. Actually, there are way more applications for the systems than I showed you today. If you have any question upon your application, just write me an email, or you can ask a question right now, and we will try to answer them and try to find the best solution for yours. Thank you.
Okay, Robert. Okay, Robert. Yeah, thank you very much so far. So I just want to directly step into the questions. So one is related to the size of the sensor foil. So what is the minimum size for correct working of a sensor foil? Maybe you can also talk about the maximum size of a sensor foil.
Let's start with the maximum sensor foil size. It was presented in the foils with the stats of the sensor foils. The maximum size is up to 40 times 40 square millimeters. If you need bigger areas, you can also write us, because we have customized solutions where you can also record bigger areas. The minimum size of the sensor foil for correct working is, I would say, approximately 1 times 1 square millimeters, because of statistical reasons to make a good calibration and for easier handling. I think if you have smaller sensor foils, it would be hard to glue them inside your vessels.
Mm-hmm. Okay, another question, or actually two questions concerning cell adhesion. So can you talk about this? How can you adhere cells on the foil, and is there cell surface treatment?
You can, for example, use 1 minute of plasma treatment, or you can also use other substances for cell attachment facilitation, such as fibronectin or polylysine, or also FCS or BSA or media or extracellular medium. So you can cover the sensor foil with a very thin layer of these substances, and then the cells adhere very good to the sensor foil.
Then another question concerning is there an influence of ambient light to the measurement?
Actually, we are measuring light signals, so there is an influence of ambient light. The VisiSens system has several attempts to protect the signals from ambient light. First of all, there are some tubes that you can screw on top of your detector, and these tubes hinder ambient light from coming from the sides to be detected by the detector, and they also have the benefit that you have an adjusted field of view, and you can always have the same pixel resolution in your experiments. On the other hand, the sensor foils are covered with an optical isolation, which additionally blocks a certain amount of the ambient light. But I personally would recommend, for the best results, to use it in the dark state, or if you use it in the incubator, it's dark anyway. Dark state would be better.
Okay, then about the resolution, or let's say the working principle, how can you quantify the oxygen in the center of a scaffold?
If you want to quantify the oxygen in the center of a scaffold, there are several possibilities. As we're measuring a two-dimensional planar oxygen concentration, you can, for example, cut your scaffold into half and place it on top, and then you get the oxygenation in the middle. But actually, most of the people would like not to cut their scaffolds. We also have microsensors that you can directly place in the inner parts of your scaffold. Let them there and do online recording of the oxygenation. But that's just at one point. So you can, for example, combine both. You can punch in the microsensor to measure the inner parts online, and you can measure the oxygenation of the cross section of your scaffold at the same time, for example.
Okay, Robert, I just realized we are basically running out of time now for the scheduled time. So thank you very much again. We will continue to answer questions. Robert will send an email to all of you, including a link to the recording of this webinar. And he will also be able or be available for further questions. Just reply and send them by email. And also, we don't want to end without saying that this system is available for evaluation. So if you are interested, then just contact us either through Robert or through our website, and we will arrange an evaluation of the system. Thank you very much for attending the webinar, and see you the next time. Thank you.